Hi there. Hope everyone is doing well. My name is Tony. I am from the NGS Academy team here at UCT. Professor Nikimoto is our PI, and I'm just going to take you through the training curriculum that the NGS Academy has been developing together with the African CDC. So, in many of the low-income countries, we have a number of issues with bioinformatics, pathogen surveillance, and the NGS Academy was essentially established to run a number of training courses across the continent to sort of enhance and improve pathogen surveillance to create more capacity for bioinformatics. Since the subject is quite broad and it's not very straightforward and easily understood, we have been working closely with the African CDC to try to create a standardized document which outlines and gives an overview of the different pathogens, the different training topics within the subject of bioinformatics to give a better guidelines to individuals and institutions that essentially wants to learn about the subject matter. I mean, just over a decade ago, there were only a handful of institutions within Africa, the continent, that had bioinformatics analysis in terms of the capabilities, engineers, wet lab, and other scientists. And since the COVID pandemic, it has really driven a need to really establish high institutions that's able to produce sequencing data and able to analyze it. So the document that we have created essentially has a table. So as you can see at the bottom here, there are different tabs. So essentially the generic course, we would like to utilize this for institutions and individuals that's wanting to sort of come into the field of bioinformatics. So whether you are an institution team that's trying to increase your sequencing capacity, you have different individuals on your team, this generic course overview should give you a broad perspective in terms of the different topics that needs to be understood and the competence that's needed for each individual in your team. And then subsequently, you can see we have different subjects. So there's a viral curriculum, there's a bacteria curriculum, there's parasite, there is a fungal curriculum, and the AMR, which is antimicrobial resistant, and then lastly, a malaria curriculum. So obviously, depending on which pathogen that you're currently working on, each of this curriculum further subdivide and highlights the topics that's important for that specific pathogen. So let me first take you through the generic course. So as you come into this spreadsheet, this is an overview. It's a summary of the different topics that's required. And you can see that these are color highlighted. And then right at the end, the blue color is for essentially subjects for the wet lab. So people that's in the laboratories performing the DNA or the RNA sequencing. And then the pink color is for the pure bioinformaticians. So these people are running workflows. They need computational skills. And then essentially the green color is subjects that's sort of overlaid between both wet and dry lab individuals. So for example, the first topics is health and safety protocols. So this will require individuals to have some level of understanding of personal protective equipment, how to handle themselves in the lab, and then that's the content that will be covered. And then the course objective itself, you can see here. And then the lab practice. So essentially, again, for DNA, RNA sequencing, what is needed, what technology that you're going to need, and also the theoretical and practical training knowledge that you need to operate these machines. And then, as you can see, so these are highlighted blue. So essentially, it only for people that's actually going into the lab. So if you're a pure bioinformatician, you're running only data on your software, then obviously those would not apply to you. And then the green color, which is applicable to both the dry and wet lab individuals. So you can see the first subject is ethics, and then the general introduction to epidemiology and pathogen surveillance. So what is genomic epidemiology, and how do essentially the different concept in disease surveillance and how this can be used. So we have made some comments here, whether it's to split this into an overview or then a more modularized detail. And then you can see the subsequent topics: study design, introduction to NGS. And then once you have the result, how do you present this to inform policymakers and essentially present this result to different stakeholders? As individuals come from different levels of knowledge, you can't use technical jargons from one group of people to the next. And then the last category, which is highlighted pink. So these are more sort of pure bioinformaticians. So you will need to essentially have computing programming background, whether it's scripting, running on command lines, data analysis, then phylogenetics, theory, there's workflows, containerizations, and then as you can see, it's either Python or R, depending on what software you're using, and then subsequently GitHub version controls, and then how to sort of use this to standardize your results. So that is essentially the general course module. And then you can see under the virus curriculum. So this will essentially be for people that's running a pathogen onto a virus. So if you're working on the, for example, in bacteria, then obviously this virus curriculum might not be as helpful for you. So you can see some of the module content here. It's a C generic course, which means we have essentially covered some of the concept here, but then over here we might include some topic that's specific to viral pathogen. So that's why we sort of subdivided these topics. And the color sequence is essentially the same here. We have the green and then the blue. And then you can see for the topics we have made it more specific now to the viral pathogen. So there's technologies like Illumina, Oxford Nanopore, ONT, there's Nuclei, Pendulum, and then the Galaxy workflows. And obviously, depending on what you're using, what machines or methodologies or workflows or pipelines, you can obviously select these modules individually. And then this will obviously be covered in that module content. And then the phylogenetics and the data submission. So these are similar to the ones we had in the generic course. So we essentially just put that here. So it is the same structure and layout for each of the other pathogens. Most of the topics that's covered in the generic course, we will essentially include it here to say C generic course. And then obviously there will be topics or modules that's very specific to this pathogen. So essentially we would include that topic only in that pathogens sort of table. And then the parasitic one, as you can see, and then fungi, and then the last two are the antimicrobial resistance, and then the malaria topic. So that is the first part of the sort of table of content. With this, whether you're individual or institution, you can come here. It gives you a general overview of what topics that's needed. And then obviously, depending on what pathogen you're working on, you can look at the specific topics and decide on the competencies that's needed for the members of your team in order for them to run the specific workflow pipeline. The second part of the sort of curriculum is this more detailed document. So over here, we essentially want to give people the opportunity to go through this document, and this will essentially serve as a guideline in terms of if you want to run viral pathogen surveillance, this curriculum will provide all the knowledge and all the competency someone would need essentially to take that data from getting it into lab all the way through to analyzing it on a computational point of view until you get results, and obviously how to present that result. So we have divided this document into several sections. So there's an introduction which says the aim of this curriculum is to give an overview of the viral pathogen surveillance using next generation sequencing. There's different technologies that you will see that is introduced later on, and then there will be different elements that's covered, such as the legal, ethic, and social implications of the work, and then the scope of the virus pathogens curriculum. This is in terms of the workflows that's going to cover the data processing, different analysis. And then over here, we basically have introduction, intermediate, and advanced. So this is essentially how the modules or the difficulties of the different modules, how it's broken down. So you can see for the introductory, it's basically just relevant content, a basic understanding from a fundamental level. And then from an intermediate level, you might need to be able to be more in depth in terms of applying, evaluating the outcome. And then obviously, if you're at the advanced level, then the participants should be able to analyze the existing tools and design and implement processes that's better and more effective. So this essentially exists for us to sort of give the individuals guidelines when they come into the start. Because if you are working, if you just started, then you can look at the sort of more introductory modules. If you have been working in the pathogen surveillance space for maybe 2-3 years, you might want to, start looking at the intermediate level. And obviously you have worked in the field for a number of years, you are thinking of sort of tackling at the advanced level. So if the modules would cover the resource that's provided, you can see there's PDF presentations, lecture and pre-recorded lecture videos. There is sort of assessments for each module, exercises that you can also download, and links to some of the virtual free available softwares online. So the objectives of the training curriculum. So once again, this is only for the viral pathogen surveillance. So we would essentially have a detailed documentation for each of the other subjects that you saw on the table of content. So for example, there will be one for malaria, there will be one for fungi, and there will be a different document for parasites. So once you have completed this training, you should be able to understand the genomic surveillance, the process of monitoring the pathogen, analyzing the transmissions and genetic similarities as well as the differences, and obviously understanding some of the wet lab aspect of high quality data, how to generate this, how to obviously correlate this to the data in a specific regional community, and obviously disseminate the results to better inform policy makers. And then here's some of the learning outcomes for the viral pathogen surveillance curriculum, some important questions one may want to answer. And then the curriculum overview is essentially the table that you saw a bit earlier. And then the color code still is applicable here. And then we just essentially made it this a bit more compact. So we're able to show the topics that's covered and a bit clarification on what needs to be addressed in each of these modules. So the color code is here. And then essentially for module 1, so this is where we now would like to get the helps of individuals or experts in the subject matter. So we have had 2 essentially meetings with stakeholders such as individuals from other labs, people from the SAMBI group, as well as African CDC. With each module, we would like to break them down in terms of number of sessions, target audience, format, whether it's lecture, videos, or pre-recorded, as well as the level of these module, whether it's introductory, intermediate. And we obviously need people who works in the subject field, who are experienced in terms of they know the subject matter very well, and they know exactly if someone needs to come into this and be proficient in it, what information needs to be covered, and very importantly, the learning outcomes as well as the assessment questions. So once you have covered all the topics in this module 1, which is Introduction into Genomic Epidemiology, what are the sort of walkaways or the competence that you need to achieve, and then essentially for the assessment, how we go about to test whether someone has really completed this and understood the concept that's being covered. So you can see that we have some questions here, truth or false, to obviously test the individual. And then you can see for module 2 study design, we also essentially have the same concept, and the assessment question is not completed yet. And then for module 3 on Oxford Nanopore Technologies, you can see this module is a bit bare because obviously we are not expert in this subject, and we do require someone who is able to assist us. So as you scroll down, you will see there is a heading for each different modules, contributors. So obviously if you could, if you are expert in this field, please put your name down and start contributing to this module. Give us some description of the modules, the learning outcomes, and then the assessment that's needed to test someone once they have completed this module to ensure their competencies. And then essentially this scrolls all the way down, and each module is broken down and essentially presented in the same format. So we would like someone to obviously be able to access this freely and accessibly online. So once this is being checked by industry experts and we have completed, we would like to publish it, and so different individuals are able to access it as soon as possible. And then right towards the end, we do have resources. So if you have any publications, documentations, videos about the different subjects, please do post them here, because this will be useful for the participants to be able to access as well. Then right towards the end, we have the sort of target audience. So these will be essentially people that would come onto the course. So whether you're a public health biologist, whether you're pure bioinformaticians or epidemiologists, you can see that people do have different levels of experience and background, and that is essentially why we introduce the different learning levels, like the introductory, the intermediate, and the advanced level. So I do hope that is clear. As I mentioned, we are still in the process of creating these documents for the other topics, and we obviously hope to get contributions from you if possible. We do understand that no one is an expert in all the topics. As I said, bioinformatics is quite a broader field, and obviously depending which pathogen you are working on, you're very specific. So if you can just assist us in terms of filling out the content for that topics, we would greatly appreciate it. Thank you very much for your time. I will just sign off here.